Yeah, well, you may be sitting there wondering, why am I going to be talking about emerging list viruses and emerging virus classes? Well, hopefully that will become apparent over the next few slides. And why list virus vaccines? You have very good rabies vaccine. It provides high levels of protection against infection with rabies, but again, may not be quite so robust when you look a little bit more further afield, more divergent strains of virus that we have. So this, for those who don't know, is the sunny south coast, although we've obviously brought the weather with us today. And it's beautiful, I was told. It shines like that every day of the year. When I went from Wakefield, I thought, oh man. The only problem with that, though, wasn't true. You get mixed weather, but if anyone is a virology you like, the coast, you like a national park, that we are the only university, Sussex University, who are positioned inside a national park within the UK. And see, I'm trying to improve the virology capacity of the university. So happy to discuss that further. The one consistent thing, though, that there is on campus, once you get students during term time, seagulls. This is the top of the medical school building, you can just see here. And they are there sitting on the solar panels 24/7. Anyway, moving on to why you're here. Apart from hear about all the other great work that's going to be presented or has been presented. List work is part of what we do in my lab. The overall or overarching aims are, as part of our supertype unit, we want to develop supertype-based assays to study emerging viruses. We then, as Nigel alluded to, can use these various applications, one of those being epidemiology studies, and Kelly mentioned the here at Bat study that we were part of as well. We look for where these viruses reside in their natural reservoirs. We also like to take those outer envelope proteins, the viral envelope proteins, and tweak them, manipulate them to study the antigenicity, which parts of those proteins are perhaps the best at stimulating an immune response. That then feed into designing vaccines. We also have another avenue of investigation, which I'll touch on very briefly with Jonathan Heaney at the vet school, using bioinformatic approaches to design these improved and vaccine antigens that can neutralize more than one specific species or lineage of virus. And more recently, with collaboration with a colleague at the University of Sussex, Casanello, where we've been combining CRISPR and pseudotype technology to study what host cellular factors are important in the entry of various emerging zoonotic viruses. So I won't be talking about all of those today. Nigel didn't give me enough time, so we will be focusing on looking at the antigenicity, so how well existing therapeutics, existing vaccines in this case, provide protection against the various strains of lyssavirus that we know about within that genus. And then moving on towards the end of the talk, talking about the development of novel antigens that hope to have this slightly broader specificity. It's not just lyssavirus we work on. This is the group of viruses we work on the longest. Work with a few people in the audience, Miles, Simon, on Ebola virus. Those are the two main groups of viruses I'm interested in, or my group's interested in. Dabbled in the latter, enteroviruses, namely nipah chikungunya, more recently in Rift. Most recently, though, we've moved from simple RNA viruses, where we're trying complicated DNA viruses. A PhD student who's trying to see this like African swine fever. It's a work in progress. Maybe next year, report some results, positive or negative. But for today, it's going to be the list of viruses that we're interested in. I'm going to be mostly focusing on. Now, the prototype species within that genus is rabies virus. This family of viruses has distinctive bullet-shaped morphology with the glycoproteins or the biolambent proteins studied on the outside. And for rabies virus, this is a disease, a virus that predominantly affects communities within Africa and in Asia. And the vast majority of human cases come from contact with infected dogs. And it's thought that 50% of the human population, given our populace, countries in Asia and Africa, live at risk of becoming infected with rabies. So it is a public health concern. I noticed it wasn't on GARDA's list of the standards for high consequence pathogens, but they do sell rabies standards, and we'll come back to that later on. So there is a case for why we need to study this group of viruses. If you are unfortunate enough to become exposed and become infected, you will stimulate an immune response, both B cell and T cell. Unfortunately, that's not normally not fast enough, robust enough to be able to provide protection, and symptoms tend to develop when the virus makes its way to the nervous system. You get the initial symptoms of agitation, flu-like coughs and fevers that progress through to the stage of encephalitis. We do, though, as I alluded to on the first slide, have very good vaccines against rabies virus, one species of illicit virus. And we have good treatments that are effective if you become infected, as long as you receive them promptly enough. They have a good track record of ensuring that people hopefully won't succumb to the infection. But without these, without being previously vaccinated or receiving the immunoglobulins, the post-exposure prophylaxis, it's got a 99.999% fatality rate. It's got the highest fatality rate of any virus. So again, another reason why it's a good and important group of viruses to be studied. I've been alluding to rabies as sort of prototype species within this genus. We have, I think, there are now 20 species that I'll come on to. Here, we have a rabies virus in the top right of the schematic here. It's a radial tree using a nuclear protein sequence. And the silhouettes represent the hosts which the virus has been founded within. And within what I'm going to refer to as phylogroup group 1, this is a way that we designate viruses. We subclassify them based on genetic sequence, based on their antigenicity. Within the phylogroup group 1, you have a range of other different species: Australian bacillus, European bacillus 1 and 2. There are cases of European bacillus 1 within the UK. So rabies-free designation is correct, but be careful if you come across any bats that have fallen to the floor. Now, as we move out from rabies virus species, we get more divergent, and then we move into what we refer to as phylogroup 2 viruses. These are ones where the genetic sequence is evolved and they're distant from that rabies species. They're thought to cause the same disease once you become infected. The Lagos bat virus here in green will cause the same disease as rabies virus. Unfortunately, though, the existing vaccines, the existing treatments that are all based on this rabies virus species do not confer any protection against these phylogroup 2 viruses. And then we go a little bit further out, a little bit more divergent, about phylogroup 3, 4. And again, these are viruses where existing treatments don't confer protection. It's thought that they would cause the same disease if these viruses were to infect humans. And so all of these viruses pose potential risk to public health and obviously animal health. Those are ones that have been, or viruses that have been ratified, those are ones that have been designated by the ITCB, CPV, as official list of viruses. There are two which have been reported more recently. Matlobac virus was reported in 2020 from archived species, or archived samples from 2015 from the Tau region in South Africa. I'm not quite sure how much you can see on the screen, but it's shown here. These are the isolates of NBLV that were detected, and they sit within this phyla group 3 grouping. Just phyla group 3 virus, again, one which we don't have any particular protection against. Much more recently, a couple of months ago, and apologies, I'm about to butcher the pronunciation, bat virus, DBLV from now on. Again, from an archived sample, 2014, was detected in a myotis bat from Slovenia. However, this yellow circle here is one virus, it sits just here next to KBLV. So theoretically would confer protection. However, those studies have not been undertaken yet, but sure they're ongoing. So now we have our 20 species or potential species of listed virus. And for the topic of today's talk, I'm going to focus on two, TBLV and KBLV. Now these are ones which are working with Ash at APHA. We had a joint PhD student. You'll see Shikya Tal come up a lot. She undertook some work to characterize these viruses and figure out whether existing treatments, existing vaccine protection. So Taiwan bat listed virus. Well, actually two of them. They were isolated from skull bats. These are all insectivorous bats, and had 60–70% nucleotide identity, so enough that they could emergence. There would be a new, what was a new species of listed virus, and now the last slide, we can see that these isolates, BBLV sit within one set of viruses, and so theoretically we should hopefully see some level of protection against conferred using existing vaccines. The virus isolates themselves weren't available. However, we picked up, and what we do a lot in your labs, synthesize the genes you need, in this case the glycoproteins, and you can plug that into the pseudotyping system. But because Rebecca was at APHA, they have stuff like 4 labs, and so they could use reverse genetics to generate replication competent viruses with the DBLV glycoprotein on the surface. And we looked to see how well that virus grew. And the backbone of the reverse genetic system is the CSN. This is a isolate that is used as a vaccine. Basis of a vaccine that's taken and cloned, and it's a rabies isolate, but you can drop in and out different glycoproteins. Well, that's predominantly what Rebecca was using it for. So the top line here is the CSN, the rabies recombinant virus. It grows to titers of 10 to the 8. You can see there is a 3, possibly 4 log drop down to the TBLV titer. So it does grow. We can rescue the virus. And see from exercise, we can go on and use it. But there's obviously something there that is impacting. Maybe it's not tissue culture adapted. CSN strain absolutely is. So it's a vaccine isolate. so it's been passaged many times. And the glycoprotein we used to generate this recombinant virus was actually codon optimized. And so, to you who work with viral envelope proteins, sometimes it's good, sometimes it's bad. We think, loop back to this in a few slides' time, codon optimization didn't help in this situation. But we had the virus, Rebecca, to grow the virus. We're able to run our first neutralization test to try and determine what level of protection or what cross reactivity there might be with existing standards. Here they are, Jonathan. Here's your WHO international standard and the World Organization for Animal Health, OIE, standards, the two that we used. And if we just focus on the left-hand set of bars here, we've got antibody titer here, but the samples were diluted to specific international units. And each of these bars represents an increase in potency of those standards. And take-home message is that to get the same levels of protection you would see for rabies virus with these standards, with TBLV, you need twice as much for the OIE standard and 5 times as much for the WHO standard. So 1 and 2.5 international units per mil. So the cutoff, what's normally called the level of protection for the rabies virus, you can achieve 0.5 international units per mil. That is a whole other discussion, debate on itself. I'm just going to skip over that. So you do see protection, or do see this cross-reactivity, which is promising with the rabies globulins, with TBLV. What about looking more further afield? What Rebecca was able to do, she generated a whole panel of viruses, some of them the authentic wild-type virus, some of them recombinant viruses, was able to immunize rabbits and then isolate the blood and sera. And so we have this panel of lyssavirus sera, unique globally. I'm fairly confident I can say that represents each, well, at the time, 3 or 4 years ago, each known lyssavirus species. And we can look and use that panel to see what level of cross-reactivity there is within the different species, the different phylogroups groups. Now, based on genetic sequence, the phylogenetic tree, EBVL1, European Bacillus virus 1 and 2, and Aravan are the most closely related to TBLV. And this sort of bore out a little bit within the neutralization assays. We have those different sera that I was describing along the x-axis here, and then each of the 3 viruses. We had the WBLV, the CSN is recombinant rabies virus, and then the CVS11, the wild-type authentic rabies virus. And we just focus on the black bars. We can see here we have high neutralization when you use the Aravan sera, and the EBVL1 sera. So that is encoding that. There's sequence information can confer antigenic potential, not immunogenic potential. What though didn't match, and where you always then have to take the prediction using genetic sequences, extrapolate that into neutralization and potential cross-reactivity, is that rabies was quite divergent, almost at the other end of the phylogenetic tree, where we see good cross-neutralization, cross-reactivity with the rabies sera and WBLV. And then EBLVL2, which was closely related to the WBLV, we don't see any, or very, very little cross-reactivity. So we can take from this, there is hope, there's a potential that if someone is vaccinated, there would be some level of protection conferred by those vaccines to TB-VLB, but you just need a high level of antibodies stimulated. If we move on to the second virus, Balati virus, KBLV. This was another European virus found in, or detected, I should say, in a mycosis bat from Finland. No virus isolate was actually detected or able to be grown out from the sample. So this is all these studies now based on genetic information. But again, closely related to EBVL2, Kujan virus, and Poplobat virus. And that Poplobat virus is another virus found in Europe, in Germany. So we do obviously have these bat viruses circulating within Europe. Again, this blue shape here is one of one viruses, so theoretically we should see protection. We did a very similar set of experiments like we did before, so I'll run through them a little bit faster. Grew or regenerated the recombinant virus, it grew better. Again, this wasn't codon optimized, this glycoprotein, so that's why we think codon optimization may have played a role, but still a log or so down. Again, maybe it's got to do with not being KBLV not being tissue culture adapted. And we saw a very similar pattern with the two international standards. The viruses were neutralized, you just needed a higher level of antibodies to achieve the same level of neutralization as you would with rabies, very similar to DBLV. Looking at the cross-reactivity in that panel of this is virus sera, EBV1, 2, Kujan, and Poplobat virus, most genetically closely related. And a slightly different orientation of the graph. This time we've got the viruses along the x-axis and the sera represent each bar. And these three sera that pop out quite high above that nominal 0.5 cutoff fit with the genetic data, EBLV, EBLV1, and EBLV2. So that is encouraging for those who want to extrapolate functional data from genetic information. But we saw some level of neutralization with the Kujan sera, but not above that level that we would suggest there was a cross-protective. We had the opportunity to look to see whether existing vaccines, we're using a virology, a human rabies vaccine, would confer protection in mice against infection with KBLV. Here we can see the antibody titer is obviously a lot higher above that cutoff for the vaccinated versus the mock. And the solid line here shows that the animals that are vaccinated are protected from infection, and vast majority survive challenge with high inoculum of the virus. So actual data there from animal models showing that there is protection. But I have, as I've alluded to, while most of the phylogroup 1 viruses there is some protection, there is very little against phylogroup 2 and phylogroup 3, shown by other groups, which have been known for many years now. And there are documented cases of those non-rabies virus, viruses in humans. The exact level is unsure due to diagnostics and misreporting, but we wanted to investigate this a bit further, and I've got just 2 or 3 more slides done. We do this by 2 approaches, looking at specific epitopes and playing around with those, looking to see which ones might be most important, and then developing these antigenically optimized glycoproteins, list-virus glycoproteins, using bioinformatic analysis. So this is a schematic of the rabies virus CBS11 glycoprotein on the left-hand side here, and each of the white bars or lines here is a epitope. There are 5 main known epitopes within. We count site 2 as one site, which is conformational, 5 or 6, And because there's no cross-neutralization, antibody that binds to a rabies virus epitope won't bind to a Lagos bat virus epitope, so that's a 5-2 virus. So we took advantage of this, swap them around, and again, I will just draw your attention to the light grey bars here in the middle, these three results here, here, and here. This is neutralization assays. We took those glycoproteins, we made a panel of them, each one of those sites that had been mutated, and using a range of sera, rabies, those top two, and one was specific for Lagos bat virus. And you can see when we mutate the rabies glycoprotein to have site 2 epitope of the Lagos bat virus, you suddenly see the highest levels of neutralization with that Lagos bat virus is 0, suggesting site 2 might be important. Skip on. Study West Caucasian bat virus. This work done by Emma, who's in my group. 5-3 virus now, and take-home message when you're using a similar approach is that site 1 looks the most important. And where we're at now, I've had a number of different undergraduate MSc students generating a panel of these epitope mutated glycoproteins covering pretty much all of the file group 3, most of the file group 2. So you need a good PhD project on lyssaviruses. So we get a student to undertake final mutations and run the neutralization assays to build up that picture of what epitope is important in neutralizing which lyssavirus species, with the idea, can you combine some of these in one antigen? Finally, work that we started 2016 with Jonathan Heaney, Vet School at Cambridge, and he now is taking this idea and primarily work at University of Regensburg, Ralph Wagner at University of Regensburg, and John at University of Cambridge, using bioinformatic analysis, immunology data. They have we had a way of developing antigens that would improve that immune response that's stimulated, that would hopefully hit more than one species, more than one subtype of the virus. Again, this is data I will hopefully be able to show you next year, next time the conference runs, when the pattern has been organised. But we've done this for lyssaviruses, and it looks promising. But just to summarise, that lyssaviruses should really sit firmly within emerging zoonotic viruses that pose a high public health threat, but also a threat to animal health as well. While we do have existing vaccines, these need to be updated. There are a growing number of new species being identified, and some of these are very divergent. Chances of those providing protection being neutralized by existing treatments and vaccines are very minimal. So that's where we're at. We are now developing these optimized antigens. I'll show you a snippet of that data. I'll show more next year or to come. Just say thank you very much to Rebecca. The majority of the data I've shown today was work that she had done in collaboration with Ash, who was at or is at APHA in the rabies group, now joined in 2001, which had nothing to do with the sudden spike in avian influenza cases. And obviously Emma, whose data briefly alluded to, with Jonathan and who I collaborate with, and Simon, who have been long-term collaborators and go-to when I have questions about student time. So thanks to everyone there and funders, and if I have time, I'll have questions.